Let's understand the structure. Now Watson and Crick's model of DNA Nobel Prize winning structure अपने syllabus में है. Now this DNA structure बहुत ही difficult है इसको समझना, लेकिन इसके स्ट्रक्चर को बताने में जो कंट्रीब्यूशन है वो सिर्फ और सिर्फ जेम्स वॉटसन और फ्रांसिस क्रिक का नहीं है. Now this is a lady photo. You see the photograph. There's a lady scientist there जिनका नाम है Rosalind Franklin. Now Rosalind Franklin was, this must be in the year 1948 to 49. Now Rosalind Franklin was a physicist. She was working in the lab of Mr. Scientist Morris Wilkins. उसी lab में she was actually working to understand the structure of DNA. उन्होंने एक्सरे डिफ्रैक्शन मेथड यूज करके डीएनए के कुछ पिक्चर्स लिए हुए थे, और दैट पिक्चर इज आल्सो सीन ऑन योर स्क्रीन, एंड दोस पिक्चर्स वर एक्चुअली taken up by Morris Wilkins, was shared with James Watson and Francis Crick, and with the help of these pictures they were able to construct a 3D model of the DNA, which made them won the Nobel Prize. So Nobel Prize was shared between James Watson, Francis Crick, and Morris Wilkins. Unfortunately, Rosalind Franklin could not live at that time. She died in the year 1957. So this structure was given in the year 1953, and the Nobel Prize was awarded to the three scientists in the year 1962. Let's understand this Watson and Crick's model of DNA. Now, before Watson and Crick's model of DNA, there was another scientist named Erwin Chargaff. Now, he had given a rule in a DNA. Now, he did not discover the structure of DNA, but in DNA, remember, the number of purines will be always equal to number of pyrimidines. This information was already declared by Chargaff before the Watson and Crick's model. Now, Watson and Crick accepted this rule, and yes, it was approved, and it was considered as a fact that number of purines will be always equal to number of pyrimidines. So we will always mention as Chargaff's rule as A plus G upon C plus T is equal to 1. That is, A plus G is always equal to C plus T in any DNA. Next is the structure of DNA. Watson and Crick described this DNA as a double helix. A double helix means it is made up of two strands which are spirally coiled around each other. That's the meaning of double helix. Now, if the DNA is made up of two strands, let us understand the structure of one strand. Now, each strand is made up of many nucleotides. Now, this is one nucleotide. The second nucleotide is joined to the first nucleotide with the help of this bond. Now, this bond you can see is on the third carbon of the sugar of the nucleotide is the ester bond. It's a phosphoester bond with the second nucleotide's phosphate. And thus we can say the bond is between third carbon of one nucleotide to the phosphate of the next nucleotide. And similarly, the chain will continue making these phosphodiester bonds, and the chain of nucleotides will make it a polynucleotide chain with the help of these phosphodiester bonds. Not to forget, the last nucleotide will always have one OH group, a free OH group, because it is the last one. There is no ester bond. It is basically for the next nucleotide which is yet to be joined to the chain if necessary. So remember, the phosphodiester bond joins the or makes the chain. And don't forget, we also will always say the backbone of DNA. Now, what is called as the backbone of DNA? The DNA backbone is basically the sugar and the phosphate together is called as the sugar phosphate backbone of the DNA. The nitrogen bases do not make a part of the DNA backbone. Now, base pairing. The Watson-Crick model mentions that the nitrogen bases in the DNA between the two strands are always paired. In one nucleotide, if it is adenine, the opposite nucleotide will always have thymine into it. Guanine will always pair with cytosine. Again, thymine will pair with adenine and guanine will pair with cytosine. This is called as the base pairing. Now remember, as you notice, adenine is a purine, thymine is a pyrimidine. So one purine is always pairing with a pyrimidine. As I told you, purines are double ring compounds, pyrimidines are single ring compounds. So one double ring, one single ring makes a bond, because two double rings cannot make a bond. It will make the DNA too wide. Two single ring molecules also cannot make a bond here in DNA, because again it will be too narrow for a DNA molecule. For a DNA molecule, the purines and pyrimidines should make a consistent width of the DNA. Remember, the two strands are spirally coiled around each other. The diameter of the DNA remains same all the time. That's the base pairing. Now, there are double and triple hydrogen bonds between the two strands of the DNA. Now, for example, the adenine and thymine always show a double weak hydrogen bond. Cytosine guanine always show triple bonds. Again, adenine thymine double bonds and cytosine guanine triple bonds. This is a very important. These are actually called as the steps of the twisted ladder. That's the hydrogen bonds, and they are weak. But even if they are weak, because there are so many, it doesn't make the DNA very weak. It is quite stable because of so many weak bonds. Next is purine-pyrimidine ratio. In any DNA, as per the Chargaff rule, if you also take out the ratio of purines and pyrimidines, as it is said by Chargaff, it's equal, it is always 1:1. Both the strands of the DNA are also complementary to each other. That means if we know sequence of only one strand, can we determine the sequence of the opposite strand? Yes. Like for example, if the two strands got separated, if we know the one strand, the second strand sequence is easily determined. Here it is. For the second one also. So if it is TCAG, it will be AGGC. So like that, they are complementary to each other. Now DNA, both the strands also have some names to the ends. Each strand has a name to it. Now, if you look at the structure of the nucleotide, the sugar group there, it consists of the 5 carbons. Out of the 5 carbon, the fifth carbon location is the 5' end, and the third carbon direction is the 3' end. Now there's a correction here. Here instead of 2, it should be 3, and the 2 should be here. All right. Similarly, for the other strand, again the 3' end at the top there and 5' end at the bottom. So that makes it a 3' and 5' end in this model of DNA. He also mentions that both the strands in the DNA are antiparallel. If you see properly, both of them are in the opposite direction. So 5' or 3', the other strand is going in the reverse direction, 5' or 3'. So that's why we call them as anti-parallel. But remember, they are parallel, their width remains same all the time, but in opposite direction. The polarity. The polarity is determined by the oxygen molecule present on the sugar group. The direction of the oxygen determines the polarity, and both of the strands have opposite polarity. There are major and minor grooves. In a DNA, you can see that there are some gaps in between when it's turning into a helix. The larger gap is called as the major groove, and the smaller gap is called as the minor groove. The charge on DNA is always negative due to the presence of so many phosphates group. It's an acidic in nature. It loses the hydrogens and thus gains a negative charge. So net charge on the DNA is negative. The dimensions, the DNA structure. Dimensions. Watson and Crick have given accurate dimensions of the DNA. They have measured the distance between the two strands. Now, between the two strands, the distance is always 20 angstrom. Between the two pairs is always 3.4 angstroms. And in a helix, one helix is equal to 10 such pairs. Now here in this diagram, you can see there are 4 pairs of nucleotides. So what they say is 10 pairs make one complete turn of DNA. One complete turn makes it 10 pairs. So distance between two pairs is 3.4 angstrom. So 10 pairs makes it 34 angstroms. And 34 angstroms equals to 3.4 nanometer. Alright. So that's the length of one helix. Now one helix turns for 360 degrees complete. Half helix is 180 degrees, and what should be the angle of turn between two pairs? Now because there are 10 pairs and 10 pairs make it 360 degrees, so between two pairs it has to be 36 degrees. So that's the dimensions of the DNA. It's a quick revision of the DNA structure. Chargaff rule, you know, A plus G upon C plus T is equal to 1. Double helix, it is made up of two strands spirally coiled around each other, and it's a twisted ladder around an imaginary axis in the center. Structure of each strand is a polynucleotide chain. Base pairing A pairs with T, C pairs with G. Hydrogen bonds A double bonds T, C triple bonds G. Purinespyrimidine ratio 1:1. Both the strands are complementary to each other. Both the strands have ends, 3' ends and 5' ends. They both are anti-parallel in nature. Both of them have opposite polarity. There are major and minor grooves in the DNA. The charge on the DNA net is negative, and dimensions, as I said to you, 20 angstroms, 3.4 angstroms.